Chromatin structure is tightly coiled, so we must open it first. There are many methods to open it. One of those is its histone acetylation by the enzyme acetyltransferase, which can catalyze the transfer of the acetyl group to lysine residue on the histone tail, thus this promoting the chromatin to transcriptionally active. Next is histone deacetylation by the enzyme deacetylase that can catalyze the removal of acetyl group from the histone. As a result, the chromatin will be more tightly packaged. The third one is histone phosphorylation. With the help of enzyme kinase, it can catalyze the attachment of phosphate group to the amino acid residue on the histone tail, which then promoting the chromatin to transcriptionally active. Last but not least is histone dephosphorylation. With enzyme phosphatase, it can preventing the beginning and expression of gene by removing the phosphate group from the amino acid residue on the histone tail.
RNA processing is an important step in gene expression in eukaryotes. It involves the series of modifications that are made to messenger RNA after it is transcribed from DNA. These modifications are necessary as it can affect stability, localization, and to ensure that the mRNA is properly translated into protein. Capping is to safeguard the mRNA from degradation and aiding in its transportation out of the nucleus. The first step in capping is the removal of the 5' phosphate group from the RNA by the RNA triphosphatase. The guanylyl transferase then adds a guanine nucleotide to the 5' end of the RNA. The methyl transferase then methylates the guanine nucleotide, adding 7 methyl groups to the N7 position of the guanine ring. The capped RNA is then exported from the nucleus to the cytoplasm, where it can be translated into protein. Splicing is removing introns and joining the exons. By spliceosome, a protein complex that performs splicing, it cuts the RNA at the splice site, then release the introns from RNA transcript and join the exons. Polyadenylation is a process of adding a polyethyl to an RNA transcript, which is commonly observed in messenger RNA molecules. There are 2 distinct steps involved. First, the pre-mRNA undergoes cleavage at a poly-A site, and next, untemplate 3' polyethyl is added to the resulting cleavage product upstream.
We proceed to the translation regulations. It is the control of the levels of the protein synthesized from its mRNAs. It consists of 3 steps: initiations, elongations, and terminations. Firstly, the mature mRNA transported from the nucleus to the cytoplasms. Next, it will assembly of the initiations complex. After that, it will recognize of the start codon. Then, it will assembly of the ATS ribosomes. And lastly, the GTP hydrolysis and the initiations vector dissociations. After the initiations process, the elongations will continue. Elongations is the key step of the protein synthesis, which the nascent polypeptide chains extend by 1 amino acid residue during 1 elongation cycles. Firstly, the codon recognitions. As the codon has been identified, the peptide bond will form. After that, the translocations happen, then the acylated tRNA will be injected. It will be repeated to synthesize a whole protein. Termination occurs when a stop codon enters the S site of the ribosome. Firstly, the stop codon recognitions. As the stop codon has been identified, it will be released factor recognitions, also the polypeptide. Then the ribosome mRNA and tRNA will be recycled.
Post-translational modification is a process by which a protein structure and function are modified after it has been synthesized from the corresponding gene. Phosphorylation is the addition of a phosphate group to the polar group of specific amino acid residues. For instance, phosphorylation of serine 15, 20, and 46 activates p53 protein. Glycosylation is the addition of a carbohydrate moiety to specific amino acid residues. Glycans in glycoproteins are classified into 2 major groups based on the glycan-peptide bonds, N-glycans and O-glycans. Ubiquitination is the covalent attachment of ubiquitin to specific lysine residues. Ubiquitination of lysine 11, 27, 48, and 63 contributes to polyubiquitin functions such as degradation or regulation of mitophagy. Metallation is the addition of a metal group to specific amino acids at the N and C termini of proteins. Lysine metallation is abundant in the N-terminal of histone proteins, which regulates chromatin state and gene expression. Acetylation is the addition of an acetal group to specific lysine residues. For example, the acetylation of histone H3 at lysine 9 and 14 attracts many transcription initiation factors that contain bromodamines to the promoter.